Hello, everyone. My name is Declan Meenagh. I'm Advocacy and Policy Manager of Fighting Blindness. And you're very welcome to this webinar, which is part of an eye health series of webinars and kindly sponsored by J&J. So thank you to them. This webinar is on inherited retinal conditions. So we have some excellent speakers tonight. But before that, I just wanted to introduce someone. So Fighting Blindness has a new research manager, and that is her name is Basola. So Basola, do you want to just briefly say hello to everyone and
Yeah,
introduce yourself?
yeah. Hi, everyone. Lovely to meet you all. I'm Basola. So, as Declan kindly just mentioned, I'm the new research manager with Fighting Blindness, and I look forward with collaborating with quite a lot of you. So, yeah.
Thanks very much, and already been working very closely with you and looking forward to what we're going to do in the future on all of the research side. So, thank you to our two speakers who have given up their time this evening to talk to us. So we have Professor Julie Silvestri, who works in Belfast and is an eye doctor and researcher, and Professor Jane Farrar, who's been working with fighting blindness for, I think, a few years now, Jane, but seriously works in Trinity and, I think well known to us all, both speakers. So thank you very much. And we'll start now with Julie. So over to you, Julie. Just to say, sorry, we're using the Q&A feature. So can someone in the webinar just put in something in the Q&A and make sure it works? If that doesn't work, please email your questions to research@fightingblindness.ie. We just want to get the questions in and oh, someone has just tested the question and it works. Thank you very much. So we'll keep the chat clear just because screen readers read the chat too well. And just for some people, it's a bit overwhelming. So yeah, if you have a question, just put it in that. So either put it in the Q&A or email research@fightingblindness.ie. So with all that, Julie, if you'd like to start, thanks very much again for joining us.
Thank you very much, Declan. Can you hear me? Okay. Yeah.
Yeah.
All good. All good. Well, thank you very much for the invitation, and it's a pleasure to be here this evening. And thank you to everyone who's joined. I'm going to use some slides, and I apologize to those who can't see them so well. But it's really to keep me on track. There's not a lot on them. It's just so that I kind of stay focused on what I want to speak on. So this has been about 20 years of work that the team in Belfast have been working on the inherited retinal degenerations, essentially trying to get things ready for when treatments came. And we've had a very close collaboration with Jane Farrar and Peter Humphreys and the team in Dublin. So it's been a wonderful journey. And I think it's fair to say we are now hopefully getting to the stage that we really want to be at. So I'm going to focus tonight really on the progress in genetic testing for inherited retinal degenerations in the north of Ireland, and a similar sort of journey has happened in the south. I'm not going to do any of the tricky genetics. I'm delighted that Professor Jane Farrar is here to look after that. I'm going to focus really why I think genotyping is important, and I've always thought that, even when it wasn't just so easy to do, and then mention one of the gene therapies that is available currently and other treatments that are coming along. So basically in Northern Ireland, this was all started off with Brian Page in 1986, and then I continued on from about 2003. So we've been working in this area for over 20 years, and there's a and I'd like to give a shout out to Claire Kirk, who's our genetic counselor, and she's kindly joined us as well. And we've had a great collaboration north and south. And I guess the first thing in my mind was to find who the people were who had inherited retinal degenerations in Northern Ireland, and create a database so that when treatments came, we could find them. And for those who can't see the slides, there's a bus shelter there, which seems a really odd thing to have, but that's where we started. We didn't know who people were. We had no database. Some people didn't want to come forward for obvious reasons, and we put adverts everywhere, even on the bus shelters, and slowly it built. So we identified patients, the phenotype, which you may hear clinicians say, that is what the eye looks like. And there's a photograph there of a typical RP. And the problem with RP is a lot of it looks alike. And Professor Farrar, Jane, will tell us about the numerous genes that cause retinitis pigmentosa and the other associated conditions. So it's complicated. And on the other side, you'll see a really complicated slide which talks about all the different pathways. So I'm going to leave that. And really my aim 20 years ago was to ready patients for gene therapies when they came. So, and we've done really well, and I've had a lot of help. I've also had a lot of money to do this through different avenues, and we're really indebted to our funders. So why is genetic testing important? And I think firstly, we have to make sure that it is the correct diagnosis. And there are some things that look like retinitis pigmentosa, which actually aren't. And things like that are measles, rubella, other things. And if you can make a diagnosis clinically and it's not a genetic disease, then hopefully the thing is stable. There are also some treatments that are available to some types of RP if you supplement with a vitamin or a supplement. Refsum's disease, for instance, you can actually really slow down the condition. So it is a pity to miss that. The other thing is patients will often ask, when will I go blind? And it was always a very difficult one, certainly before we had genetic testing. You can sometimes tell from the family history, but now with the finesse, I suppose, of the genetic testing, if patients want to know, that information is more readily available. What I would like to know from the audience is, do people want to know that? Because I know I generally don't go into that in a huge amount of detail because I think it's very different for different people. So during the discussion, maybe we could have a bit of a discussion on that. The most important thing, I think, is family counselling. And people say, you know, what about my family? Am I going to pass this on? And that is where genetic testing, I think, is key. Because otherwise, you know, if you have a good family pedigree, a big family, and you see how the condition is moving down, you can have a really good estimate, but sometimes it's not quite right. And I'll show an example of that later on. And then our treatments available, and you know, a lot of these will be gene specific. So I think it is important, and we put really a lot of effort into this. So at the moment, carefully curated by Claire is our database, and we have 996 individuals on it, 602 families. So some of the others are members of the families. And I had a look this morning, and our positive results we have in 62%, which is probably about the average worldwide. We have about 14% either not tested or in progress, and about 24% not solved. And there is still that 24 to 30% of patients that no matter what we do, unless you're doing very in-depth testing like Jane's lab would be, that we can't really find the gene. Benefits: confirm the diagnosis, inheritance pattern, and readiness for clinical trials. So I just want to spend a moment on this, and this is a bit of a complicated slide, but the reason I show it is that the last time I showed it about 10 years ago, a patient came up to me and said, I'll never forget that family. So this is a large family that has been known to Belfast for 25 years, and 13 children, okay, and 5 of the 7 males were affected, and all the females were unaffected. Mum had passed away, so we just had the affected males. And when you look at that, there is no male-to-male transmission, and that is important because it tells you if there is male-to-male transmission, then it is a different type of inheritance. So what this is, the best guess from the professor of genetics and the professor of ophthalmology was that this was an X-linked family. It was coming from mum who had passed away, and it was being passed to the boys. Not all of the boys, interestingly, but 5 of 7. So, and it was very, very severe. And you know, the 2 girls who are shown further down with the stars, they were unaffected, but they're thinking we're probably carriers, and there was no gene known. And we hunted for this long time. And eventually we did actually find the gene after many years. And the importance is that the girls who are starred there in red, they had decided that they were going to do embryo selection. They were going to select all the boys out just because they couldn't bear to have their sons affected so badly. So we found the gene, and actually it's not X-linked at all, it's recessive, which means the chances of them passing it on were really very slim, unless someone else, one of the partners, was carrying the same very rare gene. So they were able to forget about the embryo selection and have gone on to have healthy boys. So it is really important, because sometimes you really can't tell, and that's kind of always struck with me. So the problem for us really was that we didn't have any money. And I know in England, the ophthalmology departments are quite well funded for genetic testing, and we really weren't. So we were doing what we could and testing people. And then the drug for Leber's amaurosis, Luxturna, came along, and Novartis make the drug. And they were very keen for us to find someone with this gene, as were we. So they offered us, in partnership with the Belfast Trust, £250,000 to genotype. And that seems like a lot of money, which it is, but genotyping for these eye diseases is quite expensive because there are so many genes. But we went ahead with it, and it was really good. And many of you will have heard of Luxturna, which is the first licensed gene therapy for rare inherited disorders, and it is recommended by NICE in the UK. So it's available, costs £720,000 for 2 eyes, but this is treatment for babies who are affected, or young people. Some older people have been treated, and it works well, but it is very rare. So I did say to them, look, you're going to give me this money, I may not find anyone, and unfortunately we didn't find anyone in Northern Ireland who had the right genotype for this treatment. I know that the South has found a number of patients, so we were very sad, but out of that we were able to genotype a lot of people. You can see up on the top, I think it's Mercury and Venus, the first dogs who were treated with this after it was tried. The dogs have a natural mutation or variant, so it was just a really great treatment to try. So with that, we have been able to genotype a lot of people, and I think it's been really very successful. So upcoming treatments in the Belfast Trust, we have been involved in a few, and I'm just going to mention the ones that we are involved in. There are many others. So Luxturna is available to us, and we still haven't found anyone with that variant. I believe Scotland haven't found any or many either. So it must be in pockets, which is interesting. Stargardt's disease is our most common variant in Northern Ireland, and there are some treatments. We've been involved in Stella 1, which is the natural history study, and very often some of these gene therapies have the natural history running so that the patients can be followed, the progression noted, and then treatment started. And we're delighted to be part of the Stella study, and we have six patients in, and that will hopefully translate to 50% of them, or perhaps more, being treated in the Celeste gene therapy trial. And at the moment, even though they're not moving in there, we are what's called a PICA, a patient identification site, where we can send patients to London for gene therapy if we think they're suitable and they're keen. Dragon study, we haven't been involved, but I have had a patient in the Dragon study, which is a supplement, which is given to prevent the movement of vitamin A into the retina. And it does seem to be getting good results on reduction of progression. It has some side effects like night vision problems, but it looks good. It's a tablet, so the risk is small. Night Star is the phase 3. And then we have had one patient sent with Usher's type 1B to London for gene therapy. Didn't go ahead, but she was suitable. So Bardet-Biedl, there are trials for Bardet-Biedl, and they're two-pronged. One is to treat the hunger and the obesity, and there is an approved treatment for that. And then the retinal degeneration part of it has a separate treatment, and that is because it's a syndrome, so it's a complex disorder. So basically, that's kind of most of what I wanted to say. For the UK, Moorfields, Oxford, and Manchester would be, I suppose, the sites that would be most sort of active in this area. And I was delighted to see that AI pulled up Belfast Trust. So that made me feel better, that at least we're getting some recognition. So I want to thank all my collaborators, all the patients that have taken part in this, and all our funders as well. And I'd be happy to take any questions at the end. Thank you very much.
And thank you, Julie. I think that's just a real whistle-stop tour, but also some real hope there with some of those trials. And thank you for all of your work and your collaboration. I know you've come to the Retina conference a good few times as well, and together on a lot of things. So thanks very much. So now we'll move on to our next speaker, Jane Farrar from Trinity. So are you ready, Jane?
I am. So hi, everybody. I'm just going to share my screen, hopefully. So we'll go and give that a go. So hopefully it should be doing that. Are you getting that or not? Hello?
It says you're at the end of the slideshow.
Sorry? Okay. Strange. I'm going to stop sharing and try again, okay? So bear with me. Okay. Okay. Okay, we're going to go again. Hopefully it'll work this time. We'll see. Okay. How's that, guys?
Yeah, I think
is that it?
Yeah.
Can you see that? Okay, brilliant. Okay. So firstly, thank you so much for inviting me to chat this evening. I just want to cover a little ground. Firstly, everything I talk about is really nothing to do or very little to do with me, and mostly to do with lots of teams we work with, scientific teams, clinical teams, patients, families fighting blindness. And in this first slide, again, I apologize for having slides. It's just pictures of the clinical sites and of Trinity. What I'd like to cover today is background in human genetics, just to bring everybody up to speed. I'm an educator. I lecture in human genetics and Trinity, and so just a little background. And each time I give a background, I think it's so awesome, genuinely in the true sense of the word, human genetics. Then I'm going to talk a little bit about inherited retinal degenerations and how we actually go about finding a disease or a variant, something that might cause disease. And then I'm also going to talk about the fact that quite often we find variants, but we don't know if they're significant or not. And we call them variants of uncertain significance, VUS. It's not just relevant to inherited retinal degenerations, it's relevant to every genetic condition. There are lots of VUS, and we as a group of human geneticists and clinical geneticists need to see how we can deal with that. I'll talk a little bit about the research that's happened in Trinity, and also a little bit about current and future therapies, and Julie gave some lovely summaries of some of those therapies. I thought before that I might just put a little smattering of Irish medical history in there because it's relevant to the retina. Arthur Jacobs was an ophthalmologist, and in 1819, he was the first person to describe a layer in the retina that involves the rods and cone photoreceptor cells. And it was called Jacobs membrane. So actually, the first name of the retina was Jacobs membrane, and it was an Irish ophthalmologist. I also wanted to give a little bit more history, and that is that we in Trinity did not start this research. It was Fighting Blindness. They started the research, they raised money to start research, and I was the first PhD student on the project, working to try and find disease genes in large families, a bit like the family Julie showed, and tracking down for the very first time, where is that disease gene and what is the mistake or variant in the gene that's causing disease. That money that started that work in Trinity, and now we have about 20 people in Trinity working on eye disease, was due to the hard work of Fighting Blindness, and also Paddy Byrne and Michael Griffith from Fighting Blindness, who actually sold tickets for a car in pubs once people had had a pint or two of Guinness, and they actually bought tickets for about 100 punts, Irish punts at the time, and raised over 100,000 to start the work in Trinity 30 years ago, believe it or not. So with that history, I just also wanted to talk a little bit about human genetics itself and the background, the amazing background to what we are, to all of us. First of all, you are composed of components called cells. And as a human being, you have about 40 trillion cells, unbelievably. And inside every cell, in the center, and it doesn't matter if you can see this picture or not, in the center is what we call the nucleus or the cockpit, where all the controls are. That's where your 3 billion letters of DNA code is present in every single one of your 40 trillion cells. Phenomenal. Absolutely phenomenal. Sometimes when I'm saying it, I hardly believe it, but it's actually true. So we're made of 40 trillion cells in this human body that we have. And interestingly, when we go to the human retina, which is the bit that we're really interested in, obviously for retinal diseases, is we have about 150 million cells in the human retina. Most of them are the photoreceptors that are detecting light. So we have about 140 million photoreceptor cells, and they're detecting light. They're light sensitive. So when light goes into your eye, they detect that signal and pass it all the way along the optic nerve into your brain. And those are such important cells that actually sometimes degenerate in retinal degenerations. But there are other important cells. We have 3 million retinal pigment epithelial cells that can also degenerate in retinal degenerations. And we have connections between cells called interneurons. It doesn't matter, about 10 million of those. So altogether, we have about 150 million photoreceptor retinal cells inside that human retina. And inside every cell, you have the human genome. The code, the 3 billion letters of code inside every single cell that's giving the commands to enable that cell to work well. So we are awesome, and we are truly really sophisticated biology. We have 40 trillion cells, but let's look and see, what controls those cells. As I said, we have 3 billion letters of code inside every cell. And interestingly, if we were to take that code and put it in a line, which we can't, but if we could, it's squashed inside the cell, inside the nucleus. There's 2 meters of DNA code in every cell. It packaged in what we call chromosomes. And so those 40 trillion cells have 2 meters of DNA each inside every cell. If you took the DNA from your body and put it in a straight line, which of course we can't, but if you could, it would go to the moon and back a few times. Hundreds of thousands of kilometers of DNA are in your one body. That's phenomenal. That's the complexity, the sophistication of the human body. So what's that code doing? And the reason it's important, because it's when we have a variant or mistake in the code that we get a disease like a retinal degeneration, or indeed other diseases as well. Lots of different genetic conditions there are. So the DNA code inside every cell is making a little message called RNA. You don't really need to know that. But it's also then giving the coding instructions to make the proteins that are really important for your cells to function. So the right code has to be in the DNA to make the right message and to make the right protein, the right building blocks for the cell. So if there's a really important gene that makes an important protein, for example, in a rod photoreceptor cell or in a cone, then maybe a mistaken protein will be made and the wrong code is there. And now the building blocks of the cell fall down a little bit. The cell doesn't work properly and eventually it dies prematurely. And that's what's happening in retinal degenerations. So DNA code inside every cell, unbelievably, is making a message and then making protein, saying that is the building block you need, or the building blocks you need for the cell. And if there's a mistake in the DNA, there's a mistake in the building blocks. What was a real breakthrough for our whole field of human genetics and for retinal degenerations is that about 20 odd years ago, we suddenly were able to sequence the code of the genome. Suddenly is the wrong word. It took lots of years of work and thousands of people working on it. And the first human genome sequence, the code of the human genome, including all the retinal degeneration genes, cost $3 billion. Just 23 years ago. Now in 2026, if you want to get your genome sequence, it's about $300. So $3 billion to $300, which is just phenomenal. So we have this human genome code, we've sequenced it in humans. And then the question really arises, are we all identical, the same? Clearly we just look at each other and we know we're not. There's variation, and we also have different diseases. We have different traits, different hair color, different height. And so actually there are small differences in the human genome between you and me. About 1 in 1,000 letters of DNA code varies between you and me. We're 99.9% the same. But now with next generation sequencing, we can sequence the whole human genome and the whole code, and we can try and look for the mistakes in that code, the variants in the code that are causing disease. I just thought it might be worth saying as well, because maybe people don't know, that when that translates through, 1 in 1,000 letters of the code are different between you and me. We have 3 billion letters of code. That means between you and me, there are about 3 million or 4 million small differences. Some of those have no effect. We call them silent. Some of them can give rise to disease, or some of them influence, obviously, our traits, our hair color, our eye color, all of those traits. When you look at the genome of even a chimp, it's actually 98% the same as a human. So we're even very similar to chimps, but we're obviously much more similar to each other. We're 99.9% the same as each other. So that's human variation, and we can see it all around us. We can see the variation in humans. But let's now focus on and consider the variation in inherited retinal degenerations and inherited retinal degeneration genes. So the first thing we'll think about, and Julie referred to this, is the clinical variation. So that's what the doctor in a clinic sees, the type of clinical variation that there is in retinal degenerations, whether the rods die first and the cone second, or whether the cone photoreceptor cells die first and the rod second, whether there's a syndrome like Bardet-Biedl syndrome, where you have a retinal degeneration but may have kidney malfunction or obesity, a number of different syndromes. So there are lots of different clinical pictures of retinal degenerations. And the 5 most common clinical pictures are rod-cone degeneration or cone-rod degeneration, the other way around, Usher syndrome, which is a deafness and retinal degeneration, very early onset retinal degeneration, which is called LCA, or Stargardt's disease, which involves more the central vision. So lots of clinical variation. But then we have to ask the question, what underlies that clinical variation in terms of genetics and genes and the DNA sequence inside the code of your photoreceptors and your cones and rods. All of those cells have the same DNA sequence in there, but they have the same as people who have healthy retinas, except maybe one or two little changes. And so that's what we're tracking on for retinal degenerations, trying to say, what are the differences in the code in a patient with a retinal degeneration versus a patient who has a healthy retina? And the amazing thing is, when I started working on my PhD many years ago here in Trinity, we thought there were 3 retinal degeneration genes. One for sex-linked or X-linked disease, as Julie referred to, one for recessive disease that the family turned out to have, and one for dominant disease. We thought there were 3. But actually, we now, as of July 2027, have a count of 528 retinal degeneration genes. Genes that are associated with retinal degenerations, either as what we call on their own non-syndromic, not with any other symptoms, or as part of a syndrome, for example, with deafness or with kidney malfunction. So there are 528 retinal degeneration genes. That's why finding the particular mistake in any given patient is a big job. It's a big job to find that mistake or that variant present. But that's what we've done together with Julie and David Keegan and Paul Kenna and all of the scientific and genetic counselling teams in Trinity and the Ioneer Hospital, the Mater Hospital, the Royal Victoria in Belfast. And actually one of our key collaborators is Nijmegen, but also globally with people all over the world by sequencing patients. And we've sequenced roughly 2,500 patients here in Ireland with inherited retinal degeneration, driven by families, patient organisations, and many, many charity groups as well. And so what we do is we take the sample and then we do that thing, that next generation sequencing, that sequencing of either part of the genome, the genes, the inherited retinal degeneration genes, or sometimes the whole genome we sequence. And then we try and analyse those data. And as you can imagine, with 3 billion letters of code, there's a lot of data to analyze. So some of our findings from these studies, and there are many, so I'm not going to go through them at all in detail. I'm just saying that when we do sequence either the retinal degeneration genes, and there's now 528 of them, or we sequence even the whole genome sometimes, we have a rate at which we get a variant that seems like it causes the disease in about 70% of patients. So a variant in one of those 528 genes is present in about 70% of patients. And that's obviously really, really important. But I think what I wanted to also say is while we have that and resolve around 60 to 70% of patients, about 30 to 40% of IRDs remain unresolved even after sequencing. Sometimes those patients have only had that subset of retinal degeneration gene sequenced, so the 528 sequenced. And so then we move them on to what we call sequencing the rest of the genome. It's called whole genome sequencing. Another really important thing that I wanted to highlight is that sometimes we will get a variant in a gene and it's not been seen anywhere around the world at all, nowhere else except in this patient. And then we look and see if it's in the parents, if we have other family members. That's called segregation or family studies. But also sometimes we go back to the lab and we engineer that mutation into a little gene and we actually express the mutant protein and ask the question, is that mutant protein functional? We do what we call functional studies. In other words, we do extra work to see if that variant is causing disease or not. And so very often there is a category of variant, which is called variant of uncertain significance, that we have to do more studies on. And it's really important that we do those studies. This is an issue for every inherited disease, not just for eye disease, for cancers, for all sorts of inherited diseases. There are lots of variants of uncertain significance that we have to track on. Another thing that we have done is taken certain categories of disease. In this case, it's Bardet-Biedl syndrome. That's where there's a retinal degeneration and kidney issues, or obesity, various different issues. And we've asked, in the Irish population of Bardet-Biedl patients, what are the genes involved? Which variants in which genes cause Bardet-Biedl syndrome in Ireland? And interestingly, there are 27 known Bardet-Biedl genes, but actually in Ireland, we only have variants in 9 of the 27. And in fact, our most common form of Bardet-Biedl syndrome in Ireland is called the BBS1 gene. That's the one that causes the disease in the vast majority of Irish patients. In about 70% of Irish patients, they have a mutation in the BBS1 gene. Another thing that we found out doing these studies is that there are variants, as I said, that have not been seen around the world. For sure, and we call those novel variants, and we usually have to do a lot more work on them to show that they absolutely are causing the disease. But we also sometimes find a variant that has been shown in other countries, but it's actually more common in Ireland. So we call that an enriched variant. So for example, this is an invariant that causes Stargardt's disease. It causes a macular degeneration, the central vision, and it's due to mutations or mistakes in a gene called ABCA4. There's 1 case in Canada, there are 3 cases in Germany, 11 cases in the US, but 25 cases in a very small population in Ireland. Compare the US with 250 million people to Ireland with about 5 million, and yet we have double the number of cases. So we call that an enriched variant. So we're actually seeing lots of new things and causes of disease in Irish populations. So when you sequence a patient with an inherited retinal degeneration, and we've sequenced 2,500, and you get a candidate, a change, a variant that in a retinal degeneration gene that might cause disease, you have to categorize it into a really strict system that's called the American College of Medical Genetics Classification System. Because you want to know, is it causing disease? That means pathogenic. Or is it benign, not causing disease? Or is it of uncertain significance, a VUS, V-U-S, uncertain significance, variant of uncertain significance, in which case you have to go back and do lots more work on it. And so we have simplified that classification into 3, really. 1, benign, that is not causing the disease. The encoded protein is perfectly healthy and doing its job well. Or number 3 is pathogenic, that is causing the disease. Or the other category is variant of uncertain significance. And those variants, and we have lots of them in that category, we go back to the lab and do additional studies on and say, can we actually look at the encoded protein? Can we see what the effect of that variant is? Will we be able to actually push the information forwards so that the patient can get a diagnosis, so that we know, is that variant pathogenic, causing disease, or is it benign, not causing disease? And we have lots of tools to help us, and this is just one of them, but we use about 10 or 15 different tools that will help us understand whether a novel variant that is found in a patient nowhere else in the world may be causing disease. And a couple of those tools, believe it or not, are AI, generated tools. So they're tools where you can actually feed that variant into a system and compare it to millions of other variants and ask, does it cause disease? How does it change the shape of the encoded protein? If there's a little mistake, one little code different in the DNA, what's happening to the protein? How does that change the encoded protein? And there are lots of tools we use. So we take those views and we do lots of additional work on them. There are amazing impacts for patients of these kind of studies. Getting a specific diagnosis in almost 70% of patients, understanding the mode of inheritance and getting a gene-specific diagnosis, understanding how the variant causes disease. Then you can target your care of patients. You can also see if there are clinical trials or approved therapies for those patients. So it's really, really important that we do this job. And we continue to do this job. However, it's also really important that we focus on the 30% of patients who have had some sequence. Their genome has been sequenced in part, at least, but actually we still haven't found a variant that might be a candidate to cause disease. So we need to sequence the rest of the genome in those patients. We need to do whole genome sequencing in those patients. And we need even better ways of understanding variants of unknown significance and doing studies to really understand what's happening. Why is that important? So I'll give you an example of why it's important. There's one or two in the literature for Lux-Turner where a patient had a novel variant in the gene causes RPE65, which is the type of retinal degeneration being treated by the only approved gene therapy, Lux-Turner. But that patient would not have been able to get that therapy until functional studies were done, and the variant that had not been seen anywhere in the world was engineered into a gene, and the encoded protein was tested. And actually, when they tested the protein, they showed it had no function. It had lost its function almost completely by comparison with the normal protein. And now the patient could be treated with Lux-Turner. Very, very exciting, very important. The reason we need to do those functional studies. So very quickly, in the last 2 or 3 minutes, I just wanted to talk about the therapies that are approved. We've already mentioned Lux-Turner and in development. And the approved therapies is Lux-Turner for RPE65. In this slide, and again, there are very little details on these slides, so don't worry if you can't see them. It just gives the name of the disease and the company that runs or has that therapy. In this case, it's Spark or Roche for Lux-Turner. And there are also 2 therapies for dry age-related macular degeneration that have been approved. When we're thinking about gene-based medicines, very quickly, I'm just going to describe what they are. If we understand the cause of a retinal degeneration and we say this gene is causing RPE65 disease, this gene, for example, RPGR, is causing X-linked disease in a proportion of patients. If we can deliver that normal little piece of DNA, the correct code, back into the cells that are affected in the retina, then we actually have a therapy. And that's the idea behind gene therapy. It's matching our sequencing of patients, our finding variants in disease genes, with being able to deliver a normal copy of the gene to the right target cells in the retina. And so we use quite often, but not always, we use little viruses that are non-harmful. If 50% of us have been infected naturally with this virus, it's called adeno-associated virus or AAV, and it's engineered so it just carries the normal gene and hardly anything else. But it can get into the photoreceptor cells or the retinal pigment epithelial cells, the cells that you need to target in the eye of those patients. And that's what's exciting about gene-based medicines. We can use these incredible tools to get a normal piece of DNA, a healthy piece of DNA with the right code that will encode the correct protein into the target cells in the retina. That's the idea behind gene therapy. And I'm really excited to say that in Ireland, for the first time, the very first patient treated with an eye gene therapy was treated in the Mater Hospital by David Keegan and team in November 2024. So that was with Luxturna for RPE65 disease, one of the 528 retinal degeneration genes. That's why we have a lot of work still to do. But well done, fantastic to get that through, and really good effects as well, beneficial effects. And likewise, the first patient was treated in the Eye and Ear Hospital with Luxturna in March 2025. So we now have 2 sites in Ireland that can do eye gene therapy, that have set up their surgical theatres, their pharmacies to be able to deliver eye gene therapies. And now we just need more therapies to deliver, for sure, or more clinical trials, as Julie also mentioned. And when we're talking about clinical trials and therapies, there is a fantastic site that if you want to go to, it's Fighting Blindness in the US. And I've put the link in here. And they will give a pipeline of all of the gene therapy trials that are ongoing as of today. It gets updated frequently. Many of them are using that virus I talked about, adeno-associated virus, or AAV. There are many clinical trials for all sorts of forms of disease, for Usher's syndrome, for X-linked retinoschisis, for X-linked RPGR, for ABCA4, Stargardt's disease, for many forms of retinal degeneration, lots of clinical trials. And if you go to the site and you can look at it, and I think there's also an audio in the site, it'll tell you which company or which academic group are developing the therapy and also what stage the therapy is at. If it says phase 1 or phase 1/2, it's in the very first clinical trial. If it says phase 3 clinical trial, it means it's already done a phase 1/2. It looked good in the phase 1/2, and now they've gone on to a bigger trial with a larger number of patients, and they're in phase 3. So that gives you an example and ideas of the various trials. What I have on this slide, and again, if people can't see the detail, it doesn't matter, is a list of just some of the gene therapies and some of the cell therapies that are present in that site. But this is only a snapshot of a few. There are many, many clinical trials ongoing for many different forms of retinal degenerations. Not for all 528 forms, for sure, but for many different forms, there are clinical trials ongoing, which is exciting. So the last slide is to say thank you, thank you, thank you to patients, to families, to Fighting Blindness, to charities, to Retina International, to all of the charities who support on a daily basis all of our work here in Trinity, to the clinical teams all around Ireland and internationally, both in Europe and the US, to past and present members of my team, which are fantastic. So the work I've described today, it particularly involves Naomi Chatterton, Sophia Millington-Ward, Arpad Palfi, Anna Ridgway, Vilta Baivanti, Sinead Brooks, and Yulia Kashmari. But there are lots of other people. Paul Kenna, who's been involved for years, David Keegan, Pete Humphreys, Julie, who you've just heard from, and many, many collaborators all over the world. So had the joy of being involved a little bit in the Luxturna journey in University of Pennsylvania with Jean Bennett and also with Spark Therapeutics on a scientific advisory board. And so wonderful to see that. But what we need to do now is to focus on getting the next generation of therapies through as quickly as possible, not in any way compromising the safety aspect of it, but just getting them through as quickly as possible. And so thank you very much for your attention. Thanks. Thank you.
Well, thanks very much, Jane. And that was a really positive and, optimistic look at what's coming down the line. Now, we're a bit thin on the ground on questions, so I did ask people to email research@fightingblindness.ie or use the Q&A feature. So if people have questions, start putting them in. And I just want to, while we're here, acknowledge that Finbar Roach, our CEO, is on the webinar, as well as several board members. So thanks to them, thanks to our wonderful speakers, and thanks to everyone for giving up your evening. The first question we got in is, I don't know, is this within your expertise, but it's related to optic nerve damage. Someone has sight loss due to optic nerve damage caused by cancer and wants to know, is there any work being done on it? But I'm not sure, would either of our speakers have...
I'm going to actually pass that to Julie as the clinician, but I will just say that there are people working on optic nerve, and also the cells called the retinal ganglion cells that feed into the optic nerve for disorders like glaucoma, for example. And in fact, we've done a little bit of gene therapy work on that here in Trinity, but lots of people around the world. And for Leber hereditary optic neuropathy, there have been some work. But in terms of damage with cancer, I'm going to leave that question definitely to a clinician. I'm a scientist, so.
Thanks.
Thanks very much. Yeah, I guess it depends on the cause of the damage. It could be due to pressure on nerve from perhaps a tumor in the brain, in which case probably not. It could be due to medications and treatments. So nothing very straightforward, but as Jane says, there are gene therapies coming for some of the optic nerve problems, which in time might be able to help.
So another question, if I want to volunteer for gene sequencing, how do I do it? Someone has an IRD.
So there are, it depends what part of the world you're in. If you're in the north of Ireland or in the south of Ireland, in the Republic. If you're in the Republic, then you can go to the Iron Hospital or the Mater Hospital. There are clinics there. And what happens now, there was a different care pathway originally when we started. It was more research based, but now what happens now is you get a sample taken, a blood sample taken, and then they send that away to a commercial company called Blueprint to sequence those genes, and then that report comes back, and we help interpret that report along with the clinical team. And then if a variant arises that needs more work, it comes to our lab in Trinity, and we start working up that variant. For example, one of those variants of uncertain significance. Or if, for example, when it goes out to a commercial company who usually sequence, sometimes only 300 or 400 of the retinal degeneration genes, if they don't find anything, then again, it comes back to us, and we do whole genome sequencing, so sequence the whole of the genome to see if we can find variants. But Julie will probably also give her care pathway for sequencing.
Well, it's similar to what Jane has outlined. In the north, one would get a referral through either their GP or their optometrist into the hospital, and then they will come to the inherited retinal degeneration clinic. I run the adult service, and there's also a pediatric service, obviously, depending on the age of the patient. At the moment, Claire's actually on there, but we would do the panel, and we've used Blueprint Genetics a lot, and we now use several of the other UK labs, which I think test around 300, 350 genes, or a more, depending if you can actually call, the disease phenotype, what we think it is, then we can actually do a smaller panel, which reduce the cost. Now, for the 25-30% who don't have anything after that, we don't have the facility that Jane has in terms of going through the sort of more detailed, although we can ask for whole genome sequencing through the labs. But we haven't really done that because I suppose that is money limited, in the north at the moment. And I don't know what the yield is Jane, what is the yield for that extra
So the yield,
30%?
yeah, it varies really on the population that goes forward. So what kind of screening they've had before. So if they've had 250 genes or if they've had 600 genes screened before. So there's a little variation. But we get something like 30 to 40% additional candidate variance.
That's good. That's good.
Okay. Thanks for that. Sorry, I'm just clearing out some questions here because we had some tests earlier. So and just I suppose so if you're in the north, it's via Julie Silvestri, and if it's in the Republic, it's via the Ioneer and the Mater. So the Ioneer, Dave Keegan, the Mater, it's Emma Dignan.
So
Or sorry, other way
it's the other
around.
way around. So
Oh,
Emma Dignan in the Ioneer
yeah, sorry.
and then in the Mater David Keegan.
They'll both kill me.
Yeah.
So just one question for you, Jane, on the genetic link with AMD, and thanks everyone for the lovely feedback as well. Really appreciate it. So someone's asking about
Yeah, really
what kinds
interesting
of
question.
genetic work with AMD.
Really interesting question, actually. Really, really interesting. So as you can imagine, AMD is a very common disease. It's about 10% of people over 65 years of age. And so right around the whole world, there are quite a lot of patients with AMD who've had whole genome sequencing. And what we do find are that there are variants in those patients that are more common in those patients than an age match normal population. So if you take a population of patients with AMD, let's say we take 10,000, and it is those kind of numbers that are done, believe it or not, because they're usually large studies globally. And we take 10,000 patients that are age matched, gender matched, and ethnically matched. And you compare their two sequences, the sequence in the AMD patients to the sequence in the patients of the same age, approximately, but who do not have AMD. You find variants that are more common in the genetic changes in the AMD patients compared with the normal patients. So now we understand that there are certain variants that increase your risk of age-related macular degeneration. Some increase the risk pretty significantly. One's called CFH, complement factor H. That's the gene name. And there's a variant in that gene that significantly increases your risk of getting age-related macular degeneration. If you have that variant, it does not mean you're going to get the disease. It just means you have an increased risk by comparison with somebody who doesn't have that variant. And so the genetics of AMD absolutely is a really active pursuit. Really, really active. Lots of teams working on it. And in fact, we have done a little work on it as well. We are interested in patients, for example, that have macular degenerations, looks like Stargardt's disease, but don't have mutations in the gene that normally causes Stargardt's disease, which is ABCA4. And we think, oh, well, maybe they are actually misdiagnosed and they actually have age-related macular degeneration. So we're sequencing those as well. So that's one thing we're doing. But also, we're also really interested in understanding for patients who do have those variants, we actually have some cells from the retina, and we're asking the question is, how are those cells vulnerable? How do they differ to cells that don't have those variants that increase your risk? And actually, we're using retinal pigment epithelial cells. It doesn't matter. It's one of the cell types I mentioned in the retina. And we are finding little differences. So that's really interesting because they are become possible targets for therapies.
And
Yeah,
that's great, Jane. And it's actually really powerful as well that the research for rare genetic eye conditions is now being used for a more common one as well. But just
absolutely.
actually on that very topic of ABCA4, someone has said they were diagnosed with this in 1979. They're now turning 60. And are there any trials available? And I know there was one trial that had a cutoff age of 55. So maybe are anyone aware of any trials or why those trials might have those cutoff conditions?
There are trials for sure. And Julie, I think, mentioned in her talk one of the trials, but go onto the Fighting Blindness website,
Yeah.
the link. It has a list of all of the trials for Stargardt's disease, of which there are a number. But what I don't know off the top of my head is the eligibility criteria for those trials. So what eligibility means, at what stage of the disease do you need to be? What does your retina look like? And what age are you? So, but there are quite a number of trials, and I think Julie will probably add to.
Well, thanks very much. Yes, so there are a number of trials, and we have been involved in recruiting for some of the natural history studies. And the reason that the cutoff is generally around that is that the disease is often too far advanced by that time. And if you're going to have gene therapy, if you can imagine the area in the center that is quite badly affected, to prove that gene therapy is working, one has to be able to measure the good retina either side. Otherwise, after the treatment, you can't show. And very often as people get older, especially there's some very aggressive mutations. And Jane mentioned we have a number in Northern Ireland, that I think are probably on the bad end of the spectrum. So some can be quite mild and progress more slowly, and others are very aggressive. And certainly by that time, anybody actually that I had picked who was anywhere over 30, 40, 45 just didn't make the cutoff because it was too advanced. But there are some patients who will have a less aggressive variant, and they could make it. But generally, that's the reason.
But
maybe that's for gene therapy. The supplementation therapies with the vitamin A derivatives are possibly different.
And so there are the supplementation therapies, but then it's probably good to say as well that there are, people looking at therapies for patients who have end-stage retinal degeneration. Now, it's not delivering the normal gene in this case, absolutely not. But there are gene therapies. Again, it's on that website. They're called optogenetics. And what they're doing is they're saying, okay, so your photoreceptors, there aren't many left. Those light sensitive cells, those rods and cones I mentioned, the 140 million of them, there are not many left. But there are other cells that are remaining. They're called bipolar cells and retinal ganglion cells and different cell types. And what they're doing is they're putting in a little gene that makes a little protein that's light sensitive into those other cells that are still remaining. And those gene therapy trials are called optogenetics. And they are for patients who have end-stage disease. So even if the disease has progressed hugely, there are possibilities of a different type of therapy. For example, optogenetics or even cell replacement therapies. So, in parallel, teams all over the world, particularly Europe and the US, are looking at different types of therapies, not just the gene-specific therapies, but more overarching therapies that will even be relevant to end-stage disease.
Yeah, thanks for that. And I think there was another question which we answered on how do I actually get, my gene diagnosed. So again, you go to the INEAR or the Mater in Ireland. So I think we've attempted to answer all the questions there, and we are a bit over time. So I'm going to say that we'll call it there. It's important that, there's a lot going on in fighting blindness, and we really want you all to be involved. There's a support service. The research department can help answer questions. We also have the VIP network, and that's how you can get involved in research, helping to write research projects, helping to provide your perspective. So I'd encourage everyone to join that, and we'll send you some information with all this. I'd like to thank our two amazing speakers, Jane and Julie, giving up their evening to share their wealth of experience and to answer the questions as well. So thank you both. And I'd also like to thank our sponsor, J&J, for this series, and also just for everyone who's, always involved in fighting blindness, always answering the emails, always taking part, because it is about us as patients having our voices heard and having the research that we want. So thank you all. and we're going to call it there. So thanks again.
Thank you very much, Declan, for
Thank you
convening
so
and
much.
sharing.
Yeah, thank you. Thank you, everybody. Thanks.